My name is Priyanka, and I'm a research scientist at Liselo Labs, or as it's pronounced in Lesotho, Liselo Labs, and I'm based here in Hilton. So I would like to welcome everybody to our seminar, Sankatana, a novel immunotherapeutic for the treatment of COVID-19 disease. Liselo is a company focused on unlocking and growing the latent value of African innovation. And we currently operate research and diagnostic labs in South Africa, Namibia, and Lesotho. So before we start, please may I ask that we keep all questions until the end during our Q&A session. I would like to introduce our first speaker, forming one half of our dynamic duo, our co-founder and the CTO of Liselo Labs, David Jarvis. David graduated with his honors in microbiology and then pursued his postgraduate studies on the gut microflora of the ostrich at UCT. His interest then led him into telecommunications, with him presenting his work at international conferences, including that at the United Nations. He now leads a team of more than 30 researchers with collaborations in leading institutions. David is indeed a jack of all trades. Almost nothing is impossible for him. David will now give us a brief introduction to Liselo and our immunotherapy platform.
Okay. Thanks, Priyanka, for that flattering introduction. So I'm David Jarvis, CTO at Liselo Labs. Good to have you here, and those on Zoom. I'd like to start by just giving a little bit of an introduction to our journey as a lab, just to give you some context to what the main topic is today, which is around our product. Because I think the journey talks a little bit to how we got there and why we think it's a useful and a useful product to consider in a therapeutic context. So essentially, I'm going to be talking about the history. We're going to get Dr. Muti to talk about the molecular virology, and then we'll have myself again talking a little bit around our therapeutic strategy to get to where we are now. So our R&D strategy around the therapeutics. And then Sapa will talk about the case study in Lesotho, which I think is the most exciting part. And then we'll have some time for questions and answers. So just to kick off, in 2012, we started Liselo Labs, Sapa and I, largely driven by coming out of the telecoms industry and realizing we want to pursue our passion, which was biochemistry and microbiology. And since then, we've been focused primarily on building immunotherapeutics for veterinary and human disease, but also diagnostics. So in the South African context, or in African context as a whole, if you don't have accurate diagnostics, you aren't able to detect what you're dealing with, and so an immunotherapeutic is almost pointless. So we're very focused on diagnostics. I'm glad to see Dr. Gilbert here as well. He's also very focused on diagnostics. You need to know your enemy, as Sapa points out quite frequently. So we've built a couple of diagnostics and research facilities around SADC, most notably in South Africa and Namibia, Lesotho, Botswana. And we have a research facility in Angola, but we also service southern Angola for diagnostics from Namibia. So just in the last year, we've been busy. We deployed 5 new labs in 4 countries in the region. And so this team that we have with us in Hilton travels a lot, right the way through the pandemic so far, the last 16 months or so. That's on top of our R&D work. So the diagnostics work has really been an extension of our R&D vision. The R&D vision we'll talk to just now is a bit more holistic than just immunotherapeutics. And we're a small team, there's only 30 of us, so having the ability to plug into the collaborations that we have at UKZN, at Harvard, at Predicare Labs in Texas, Benga Systems in Angola, and Chiara, who is a local regional pharmaceutical distribution company, gives us the kind of gravitas to do things that we wouldn't be able to do on our own. And we also have been funded by the Department of Trade and Industry, now called the Department of Trade and Industry and Competition, DTIC. And they funded our collaboration with UCT and with the CSR. We're a multidisciplinary team. Even in this room, you have vets, you have molecular biologists, you have molecular virologists, you have IT specialists, telecoms engineers. And so it talks to the ability to approach the problem from a wide view, and not the sort of myopic view you typically engage if you're just a molecular biology team. We also have an entrepreneurial team, and that team ranges from, in terms of the management, ranges from sort of the well-published academic right the way through to the humanitarian, who I think it'll become evident just now. We're all humanitarians at some level, but some more so than others. So we have a virtuous circle, which drives a little bit of our view of the world, and it revolves a lot around technology and IT, because IT is sort of the glue that holds a lot of what we do together. And so if you sort of start at the top left, diagnostics, we're all familiar with diagnostics now. Everyone can tell you what PCR means almost. Everyone outside of this audience, obviously inside this audience, we should know. But our view is that you cannot divorce the health of humans from the health of animals, from the health of the plants that they consume and that they live around. It's the one health view, which is why we have veterinarians in our team, it's why we have agronomists in our team, and it's why we have molecular biologists and medical professionals in our team. There is no separation. And I think it's so poignant that SARS-CoV-2 is likely an emerging zoonotic disease, not confirmed yet, but likely is. And so, we need to be understanding the relevance of at least animal and human health and their relatedness. That's part of what we do. When we diagnose for customers, we're looking at animal and health and human health concerns. And when we look at our intervention monitoring, which is driven by our app, we're linking our diagnostics to things like port access control at borders, so people can validate certificates, etc. And then if you move further down on the virtuous circle to the bottom right, the One Health app really brings together the ability to empower individuals to take better care of their own healthcare, and that of the animals around them, and that of the plants around them. So that's sort of a whole One Health concept, but brought into the hands of the individual. And then the point of care diagnostics, we're all familiar with, the rapid test kits. That's part of what adds to the ecosystem of an app to allow people to test for themselves in many cases, not all cases, but in many cases, because a lot of the markets we address, there is no doctor or clinic in close proximity. So you need to have access yourself to these tools. And then that feeds back into our diagnostics or our lab-based work, which also allows us to develop immunotherapeutics based on the learnings in the field from the sort of One Health view of the world. There'll be time for questions and answers just at the end, so just jot them down. So the platform, the immunotherapeutic platform, is essentially the ability to produce immunotherapeutics targeted at specific diseases affordably. So in the African context, shall we say, or the developing world context. And so we not only have a co-patented expression system, think of it as a manufacturing process, which allows us to make these immunotherapeutics locally, but we also have the ability to manufacture them en masse. And that's important because right now, in the last 12 months, we've been able to manufacture enough to service, for example, the whole of Lesotho and increasingly South Africa. But within our research capacity, we have built the ability to start at drug discovery right the way through to how you take a product to market, essentially, through our entrepreneurial team. So it doesn't help in the South African context, certainly not during a pandemic, to discover a drug and not find a way to make an impact on the community. And we'll talk just now as to the normal route for a drug to market is 12 years in the US. We've had to do this in a year or less. And then what we're also busy with, just as part of our platform, is an expedited route to registration with SAPRA, it's our local regulator, the FDA, the Food and Drug Administration in the US, where we are starting with filing processes for various drugs, most notably the ones for COVID-19. So our overall vision for the platform is to look at the replacement stroke complementing of antibiotics, right? Replacement stroke complementing of antibiotics, because antibiotics, we're about to face the next pandemic, which is the antibiotic pandemic, or apocalypse, as people like to call it. So we have a strong vision around replacement, stroke complementing of antibiotics, and with immunotherapies, targeted primarily at pre and post-exposure therapies for animal and human, non-communicable and infectious disease. Basically, a huge section of what we currently target with our antivirals, our antibiotics, our immunotherapies that are emerging around oncology, but our focus is to see a gradual move towards a largely immunotherapeutic strategy, certainly in our lab, for those conditions and diseases. So at this point, I'd like to introduce Dr. Moodley. He's our senior researcher, our resident molecular virologist, one of our senior researchers. And he's going to talk a little bit around the molecular biology of SARS-CoV-2, relevant to the therapeutic strategy that we've undertaken.
I would like to introduce our second speaker, Dr. Joey Moodley. Dr. Moodley is a molecular virologist from the University of KwaZulu-Natal and a senior researcher at Liselo Labs. While he is laser focused on his research, his interests include, to a large extent, health and wellness and beauty therapies. However, he specializes in diagnostics, pathogen interactions, and disease epidemiology. Dr. Moodley will now give us insight to the molecular biology of SARS-CoV-2 and its relevance to therapy.
Good morning, and thank you for joining our talk. And as David has mentioned, we basically look at target drug therapeutics, and in order for us to develop these target drug therapeutics, it is important for us to understand the molecular mechanisms that drive disease. So we focus today on SARS-CoV-2, and so I've got some very interesting research, very up-to-date research about the immunopathology of the disease. And so this is the basis upon which we have developed our immunotherapeutic. So if I can just get into this, this is a paper. Some of the research is based on a paper that was done by these guys, Zhang and Shai, and they are Salk researchers associated with the University of California. And so they have made some they made a very important discovery about the way the disease, the immunopathology of the disease progresses. So, so basically what they are saying is that SARS-CoV-2 is actually a vascular disease and not fundamentally a respiratory disease. It leads to a respiratory disease, but what we need to understand is that it is fundamentally a vascular disease. And I'm going to explain to you how this is actually happening. So SARS-CoV-2, we all know that there is, it binds to the ACE2. So we have your RBD, which is a receptor binding domain on the virus, which is located on the spike protein. The spike protein is made up of two subunits. So on the one subunit, you have your RBD, and that is involved in the virus attaching, where the virus is actually able to attach to your cells. So basically, and there are ACE2 receptors, which then facilitate the binding of the virus to your cells. That's how infection occurs. So, so what happens here is that what they are saying, what these researchers are saying, is that the spike the binding of the spike protein is the S1, the RBD to the ACE2 is enough to cause vascular damage without the cell actually infecting, going into the cells and replicating and causing infection. So what they did was they made a pseudovirus. So pseudovirus is basically a virus-like particle that just has the spike proteins on it. So there's no, it doesn't have the actual nucleic acid, which facilitates replication and all the other processes in order for the virus to then move and infect other cells. So what they found was that when the RBD binds to the ACE2 receptor in your lungs, then what happens is that your what you get. Okay, so this is so basically it's a pseudovirus. So it has the spike proteins, as I say, it binds, and what happens is that that S1 subunit where the RBD is located triggers this cascade of reactions, particularly where you're getting mitochondrial fission or mitochondrial fragmentation. And what usually happens in this case is that it starts to cause inflammation in the tissue at that very, it's just from binding alone, without any infection happening. It's simply just attaching, and what you're getting is mitochondrial fragmentation, and then and so then that leads to ubiquitin proteins being produced. And when you get these ubiquitin proteins, what those proteins are actually doing is it is marking your cell for death, and it goes it triggers this caspase pathway, which then leads to apoptosis, okay, which is basically the cell death, right? Okay, so in your endothelial cells, you will find that there is something called nitric oxide, which basically regulates a number of different processes during inflammation. So when you have so there's an enzyme in the vascular tissue in your endothelial cells, sorry, which then basically produces this nitric oxide, and this is it's an enzyme, so it's endothelial nitric oxide synthase. So if I could just point here, your ENOS. So what happens is that it becomes impaired. So when you have an impairment of this enzyme, you find that nitric oxide is reduced. So the moment nitric oxide is reduced, it tends to affect the regulation of the vascular tone. So what's happening is that you're not getting relaxation or constriction of the blood vessel. And so this is obviously a big problem, because you're not getting a proper flow of blood, and also your immune response and all of those things are impaired. So what happens now is that what it also does, what nitric oxide, what it also does is it has anti-proliferative effects. So when you have proliferation of cells where there's infection, you're going to find that their smooth muscle cells starts to increase. They also get an increase of fibroblasts, and you get an increase of your local immune response. And so if you have a reduced amount of nitric oxide, what happens is that it is not having that anti-proliferative effect. So it's just increasing and increasing, and it's causing this inflammation and tissue damage. And so that's the other property. And then it also has anti-thrombotic effects. So if you see with a lot of COVID cases, people start to develop thrombosis, and this is because what's happening here is that your smooth muscle cells along the are then becoming damaged, and so basically what's happening is that there is exposure of collagen and other factors, adhesion factors, and so then with the nitric oxide being reduced, you are having thrombosis occurring. Okay, so blood vessels are activated. Another very important thing to consider is that it inhibits NF-kappa B, So NF-kappa B basically regulate, if it is not regulated properly, what happens is that it then starts to get into the nucleus, and it starts to transcribe all of these pro-inflammatory hormones, and this is where the cytokine storm that you see occurring is basically regulated from NF-kappa B. Okay, so so then we also have the superoxide dismutase, which is an antioxidant that cleans up the reaction oxygen species. So it needs to mop this up. As you get a buildup of this, you start to get a whole lot of other issues that will start to occur, right? And this is also exacerbated by an increase in glycolysis. So when you have a breakdown of glucose, what happens is it then starts to increase and produce more reaction oxygen species, and then you have an increase in inflammation. So so now you will see that it tends to upset or destabilize the balance between your ACE1 and ACE2 receptors. And so when you get this imbalance due to the buildup of reactive oxygen species, then what happens is that ACE2 is actually pro-inflammatory, and so it basically then causes an increase in inflammation. Okay, so so then then it then starts to cause issues with your blood vessels, your bleeding, and all sorts of things like that. So so the take-home message from here is that this is it's a vascular disease primarily, and caused by infection, caused by the actual spike protein itself, right? Not really from an actual replicating virus particle. It's just that spike protein that triggers off this this vascular disease and causes this whole cascade of reactions, and then giving you all of these inflammatory responses, which they can have very adverse effects without the virus actually replicating. And so vaccines will help to reduce spike-induced vascular disease. So by using vaccines, it will help to reduce the spike-induced vascular disease, because the antibodies are based on those spike proteins. Okay, and so this is the article from which I've extracted a lot of the information. And so if people are interested in reading this, you can just go through that, right? So if I can move into the next part of the talk, the SARS-CoV-2 spike protein subunit S1 induces COVID-19 like acute lung injury. So basically, in the study, what they did was they are looking at just the S1 subunit again. But what they are saying here is that it is the S1 subunit alone that is involved in the cytokine storm. And this is the first study where they demonstrated what's actually causing the cytokine storm. Right? So what they did here was that what's actually happening is that, so your spike protein is made up of two subunits, the S1 and the S2. And so as it binds, when you get cleavage, when I talk about cleavage, it is basically cutting between this between the S1 and the S2 by TMPR, SS2, and furans, which are proteins located on the surface of the cell. These facilitate the cleavage to occur, and when this happens, this is actually causing the cytokine storm. So this is actually, if, for instance, the spike protein is a way to keep the spike protein from being cleaved, you're going to find that that is a target to actually reduce inflammation, the cytokine storm, and then the progression of the disease. So if you are developing the types of immunotherapeutics or drug target therapies, it can be targeted at this very at this cleavage. And so what they did was they took mice, and so they used a transgenic mice. So the transgenic mice basically had humanized ACE2. It's not mouse ACE2 receptors, but humanized ACE2 receptors. Then we have so there were different treatments. There's 4 different treatments. For the first treatment, it is just it's humanized ACE2. So it's a transgenic mice, but then what they did was they used recombinant spike protein, which they had actually had made by one of the companies that they listed in the article. So basically what happens is that they administer the spike protein to the transgenic mice, to the transgenic group of mice. In the second cohort, there is it's also transgenic mice, but they are administering the spike protein with enzymes that stop the cleavage. So it's basically a blocker of where it will cleave, right? And then in the third one, it's a wild type. So the wild type is basically where it's mouse ACE2, but they also still going to administer the spike protein that can be cleaved, right? Remember we said if it cleaves, it can it's where the disease, the cytokine storm is occurring, right? So they are evaluating what's going on. And then in the third cohort, what's happening here is that it's a control, they're just using saline, right? So then what happens is they expose the trachea, and then they put a catheter through down the trachea to administer the different treatments. So what we are interested in is looking at the first treatment where they are using the spike protein that can be cleaved, and then the wild type where there is the same spike protein that can be cleaved, right? And then so then what they do is they had observed the progression of what was happening with these mice, and what they found was that in the first cohort where there was cleavage occurring of the S1 and S2 subunits, you were getting those mice lost a lot of weight, right? Compared to all of these other ones here. Nothing happened, not much happened to the rest of them, but where there was cleavage and humanized ACE2, so in the transgenic mice having the human ACE2 receptors, they found that those mice lost a lot of weight. So if we move on, we will look at, so what they did was a bronchial lavage, which is a wash. So when they wash through you get an idea of what is happening in the alveoli, right, of these mice. So for if I have to look at the alveoli of the last 3, that would be more or less what we're looking at here. So this was more or less what the results were from there. It wasn't really damaged as much, but from the transgenic mice having the humanized ACE2, where that spike protein was able to be cleaved. So you're getting so edemas were occurring. So that is where the lungs are filling with water. Okay. So septal thickening. So basically, it's where the walls of the alveoli is becoming thick, and then that's basically affecting your gaseous exchange. So oxygen is not being taken up effectively, and CO2 is not being exchanged effectively. So then you're getting hyaline membrane, which is basically shows up as damage because of protein damage. And then so you would see this on a histogram where there are these red spots that will start to occur. So you're getting highly membrane, and then you're getting the leukocytic infiltration, which is basically your local immune response. So you're getting influx of the immune response, which is happening, which is then telling us that there is inflammation happening here based on that binding. So then if I could explain the next part of the results, what's happening is that the transgenic mouse, where we had the spike protein that was able to be cleaved, was damaged. The most damage was seen in that, as compared to the one where the spike protein could not be cleaved by enzymes. So it's just the S1 and S2 subunits stayed intact, so it could not be cleaved. And then where they used saline, or there was no lung injury seen. And in the wild type mouse, there was some inflammation that occurred, right? So although it's not a human ACE2 in there, it's a mouse ACE2, there was still some damage that occurred, right? So here you can see the cytokine storm that profile, which they then elucidated. And so you can see in red that the transgenic mouse, where you get cleavage occurring, there is an influx of those particular cytokines, and which is associated with COVID-19, this COVID-19 cytokine storm. Okay, and then if we look at the histogram here, the most important one is looking at the transgenic mice that were given the where the S1, S2 subunits could be cleaved. And what you can see here is that it's very dense. So it should be, if you're looking at the other three, they are a lot more porous. So it should be very thin, but by this being so dense like this, what's happening is it's showing all of so the cytokines, you're getting the septal thickening and all sorts of things like that. That is then causing this the lung to look like this. So you can see tissue damage occurring here. Okay, so basically from the study, what they are saying is that the take-home messages. So this is the first demonstration of COVID-19 response to an essential virus-encoded protein by SARS-CoV-2 in vivo, and this was demonstrated in mice, of course, not in humans. This model of COVID-19 induced ALI may assist in the investigation of new immunotherapeutic or new therapeutic approaches for the management of COVID-19 and other coronaviruses. So basically, understanding these mechanisms can help us to develop novel immunotherapeutics to reduce inflammation and the progression of the disease. Okay, so if I could just go into actually looking at the variants, the COVID-19 variants that are out there, and why I'm jumping into this right now is because I just wanna highlight what's actually circulating and what's really important right now, and it's the Delta variant. So this Delta variant is basic. What's happening here is it's just mutations of the virus, and these mutations are occurring at the spike protein, right? So basically, a lot of the target, the vaccines and the target drug immunotherapeutics should be directed at this particular region of the gene. So what they are also saying is that there's increased transmissibility with the Delta as compared to ancestral strains and like the Alpha strain, which we will do much of our comparisons to. There's also an Epsilon strain right now, which is in California. It's not as prevalent, and it's also not as destructive as the Delta. So we will focus on the Delta. So some characteristics here with the Delta as compared to the other strains of COVID is that it it was shown that there is 1,000 times, the viral load is 1,000 times higher as compared to the alpha. Also, transmissibility is at least 64% more with Delta as compared to the other COVID-19 variants that they had identified. And basically what happens is that the general R0 value, which is basically a transmissibility, like for person-to-person transmission, basically if you contract the virus, you could generally spread it to up to 3 people with the other strains. Whereas with this strain, you could spread it up to 9 people, which is basically as contagious as chickenpox. So it's quite severe. And then there's also been an increase in the hospitalization rate. As compared to the alpha variant. And what they found is that it's prevalent in people that are greater than 50 years, greater than or equal to 50 years, but have comorbidities as compared to those people that do not have the comorbidities. But it's also affecting younger people less than 50 years, but generally those that have not been vaccinated. Right, so that's where the problem is. So this is just a diagram to show you how the rate of transmission of the SARS-CoV-2 Delta variant, and it is in line with that of chickenpox, which is quite fascinating. So so moving on to the symptoms, and basically if I could just draw on what's really important here is that people who can't smell and can't taste generally, which occurs with infection of the alpha variant and the other variants, what they're finding is that with the Delta variant, that's not as common as it is with the other variants. It also causes hearing impairments, and then there is, in some cases, they have found that people develop gangrene. So, there's more research that needs to come out where we can really, associate that symptom with the new Delta variant. Okay, so then quickly, I'm just going to go onto treatment. There are monoclonal antibodies that you can use for patients that are not having severe symptoms but have a high probability of where the disease can progress. So they recommend using monoclonal antibodies. However, the efficacy with the different variants can vary. And so with the Delta variant, you can find that it's not as effective. So you may want to consider some alternative therapies. And so what they do is they can treat you with steroids like dexamethasone, or you can use well, basically you would want to vaccinate, right? That would be your ideal thing to do. But if you do develop symptoms, you then want to look at this at the severity of the disease, and then you administer the treatments based on your symptoms. So if you are not requiring oxygen and all of those kinds of things, you can use dexamethasone or remdesivir to basically reduce inflammation and reduce spread of the virus. If you find that your patient is needing oxygen and CPAP and all of those kind of things, you then you got to make your decision based on what the criteria that these people have highlighted. Okay. So in summary, what I will say is that the Delta strain is highly contagious and likely more severe, but then the vaccines are very, very effective at reducing severe disease and hospitalization, but not as effective at reducing infection and spread. So got to get vaccinated. And so, yeah.
Okay, so sorry about that. We present in different ways, Joey and Saf and ourselves, so it was a bit complicated. So the global strategy around COVID-19 therapy at the moment, we all pretty much understand. We have vaccines. Vaccines are your best strategy at preventing severe disease. How do they work? Immunity is built through an antibody-mediated immunity and a cellular-mediated immunity. It's pretty straightforward. We've pretty much all seen the diagrams. We understand that. But on top of that, for 90% of those on this continent who haven't been vaccinated, they are likely to progress to severe, more likely to progress to severe or moderate disease. And so we're using corticosteroids to reduce inflammation in a clinical setting. And in South Africa, dexamethasone is our go-to drug, right? Primary mode of action, limiting the production of pro-inflammatory cytokines and upregulating anti-inflammatory cytokines like IL-10. We understand that that's a strategy, and we have very few other strategies that are emerging, which allow us to do much more beyond that. Our formulation is focused on something very, very similar. It's not a vaccine, right? But its mode of action works through viral inhibition, through antibody-mediated immunity. There are antibodies in our formulation targeted at SARS-CoV-2 receptor binding domain. Okay? Much like when you take a vaccine, your antibody response is targeted to the receptor binding domain of the vaccine spike protein, which was injected into via various vectors. The other key element is NF-kappa B dysregulation. So NF-kappa B is a transcriptional factor that influences many important physiological functions in healthy cells. But in a disease state, it becomes dysregulated. So it's out of control. And in that dysregulated state, you get this pro-inflammatory and downregulation, upregulation typically in a disease state. And so dexamethasone, for example, is down-regulating that activated state of NF-kappa B, and you are seeing a reduction in the cytokine storm, and you're seeing better clinical outcomes for patients. We have NF-kappa B inhibitors in our formulation, which achieve something similar via an oral mediated application. Joey spoke about the S1, S2 cleavage, so S1, S2 uncleaved, not likely to induce severe pathology. S1, S2 cleaved. It's cleaved by a protease. It's cleaved by furin and the TPRSS1 protease. Those are serine proteases. We have serine protease inhibitors in our formulation. And then we formulated that as a food for oral administration so that its targeted sites of action are in the GI tract, mouth, esophagus, small intestine. And we've achieved that by putting it into a yogurt, which has been well published to be a mucoadhesive, but to also allow for gastric transit. We wanted to have the ability to deliver antibodies in the small intestine, where much of the infection is likely to happen for SARS-CoV-2, obviously beyond what's happening in the lungs and the oral mucosa. We also did a honey-based syrup, so I don't know how to get rid of this Zoom thing at the bottom here. Put it up there. And the honey-based formulation is also mucoadhesive. It also assists with gastric transit because of the high fructose and sucrose content. But most importantly, it's also stored at ambient temperature, which is a problem for our yogurt in rural settings. So how can we possibly be claiming that we have a therapeutic that might work for COVID-19 when it normally takes 12 years to take a drug from preclinical in the lab to the shelf? We have them on the shelf already, right? When 99% of drugs that pass preclinical studies in the lab, i.e. they will inhibit viral replication in the lab, for example, SARS-CoV-2, will typically take 12 years to get to market. 1 out of 5,000 of those drugs that successfully pass some preclinical trials get to market. Right? So we're either doing something brilliant, right? Our team must be absolutely fantastic, or we got lucky, or serendipitous, as Joey prefers, or we've worked really, really hard during the last 12 months to try and achieve something. Or perhaps, and probably more likely, a combination of the above, right? Because we've gotten our product into thousands of COVID positive patients in Lesotho in the last 12 months. And we've been able to observe the outcomes for those patients in clinical settings, which includes COVID wards in Lesotho, which Saba will talk to. Obviously, observational studies don't tell the story, the full story. They're not a double-blind, a placebo-controlled double-blind study, which is why we have those under ethics evaluation right now from Lesotho and from South Africa. And those will hopefully go into studies which in the next 6 to 12 months will yield results, right? We are registering it as a Schedule Naught, basically a herbal remedy, just so it can get to market with SAPRA and the FDA in the States. And we have already started producing the syrup and the yogurt for commercial sale, which you'll get some of just now, in about 40 locations, mostly in Lesotho and increasingly in South Africa, including the Spa and Camperdown. So I want to, at this point, just point out that the reason why so many drugs fail from lab to getting to the shelf is that the clinical evaluation phase in patients is something that we poorly understand. There's too much at play. It's so easy for a drug to have side effects which are so adverse that the benefits are not outweighed by the disadvantages, where the risks in certain groups are just way too high, so the drug doesn't have broad applicability, where the drug has other interactions in vivo in a patient which just weren't observed in the lab. So the real take home is anyone can come up with a new concoction that may work in the lab. The proof of the pudding is how safe is it and how efficacious is it in clinical patients. And we will only be able to tell you that with a peer-reviewed paper in about 18 months' time, if we're lucky. In the meantime, it's being used as a food in Lesotho, and I want Sapa to just tell us about that story so we can understand his experience.
After a detailed look at the molecular biology of SARS-CoV-2 and the science behind Sankatana, a real-life view will piece the puzzle together. I would like to introduce our next speaker, the other half of the dynamic duo, our co-founder and CEO, Sehapa Moiletsi. Sehapa is a biochemist and seasoned entrepreneur with investments in logistics, telecommunications, agro-processing, molecular biology, and the list goes on. Sehapa now sits on the committee of New Grains SA and has capacitated the Lesotho Department of Agricultural Research with innovative climate science in interventions. As COVID-19 reared its head, Sehapa rose to the challenge. He is Sankatana himself and can offer us reality through his eyes and maybe some magic. Sehapa will now speak to us about the Lesotho Sankatana case study, immunotherapy for COVID-19 disease.
Good morning, ladies and gentlemen. My name is Sehapa Moiletsi, and I'm going to tell you a story many of you are going to battle to believe. That initial slide pretty much just summarizes the picture of Lesotho in terms of the official stats. The important number to take away is that it's a population of 2.1 million people. Dave, it's not showing the window I'm expecting. Right, okay. Right, it's a population of 2.1 million people. But if you look at the global death rate sitting at 4.5 million, that means the entire population of Lesotho will be wiped out twice over, and for the unlucky sorts, 3 times over. That to me sounds like a plot out of a movie like Avengers with Thanos attacking. And it's petrifying, to say the least. Right. So what have we done in Lesotho? In 2020, in April, we were invited by the then Minister of Health, to help with the COVID strategy. And we ultimately wound up having our first case being to assist the Litsen diamond mine, which pretty much contributes the bulk of Lesotho's economy. And we essentially developed interventions and strategies and protocols which spanned everything from PCR testing with results the same day to AI tools. Basically, when we pitched what we were going to do as an intervention to those people, I remember the one doctor who's contracted by the mine basically sit up and protest and say, hey, you guys are going overboard. You are bringing an AK-47 to a stick fight. And I remember David at pains to tell him that, doctor, this is no stick fight. We will need every single weapon we can muster. We will need ICBMs because many, many people will die and many, many Basotho will die. So if you look at that number of 4.5 million people dying, it was a haunting prophecy. Anyway, we also then provided services to the general public a little later through a hospital called Wheatley's Hospital. And there at peaks, we would literally process over 900 people per day. But I can tell you this is the boring part. And fortunately, I've only got 4 slides and I've already gone through 2 of them. Because what I'm really now going to tell you is, yeah, I get goosebumps all the time. So here's a key question. Does Sankatana really work? Well, like David alluded to earlier, we've gone to great pains to roll it out as quickly as possible. And this happened mid-July this year. Granted, for the past, since July, we've been giving it to people who test positive through our lab. We make them sign consent forms and off we go and observe them, as David alluded to earlier. But it wasn't until Tepeltola died. And for those who don't know who Tepeltola is, he's a musical artist who's like the best and biggest export Lesotho has to South Africa and the world. And when he died, it was a seminal moment for me. I said, this is somebody I know. He dated my sister and now he's dead. I could have possibly done something because we had seen the most amazing miracle for a period of a year. So that's it. The shackles came off and we went to town to convince individuals such as yourself, healthcare workers, pharmacies, clinics, that, hey, listen, give your people some of that yogurt and see what happens. Long story short, in under 2 months, we basically have a presence in 9 out of 10 districts in Lesotho and at over 30 odd retailers. We've since also become part of the CCU Solidarity Fund, which is a private sector drive to raise 100 million to essentially vaccinate Basotho. Our key role in that, in terms of our contribution more specifically, has been to provide free antigen testing, coupled with, yes, you guessed it, making sure that those who test positive get some of that yogurt in them. And as David mentioned as well, we also made sure we start donating it to COVID wards. I had a cousin die. It was the first death in the family. It was frustrating for me, but let me not digress. To date, we've sold 1,750 tubs and counting. But the key thing here is that for a large chunk of yogurts we produce, we donate a heck of a lot as well. So for every 3 tubs that we produce, we donate 1, whether it goes into a COVID ward or, I don't know, impoverished communities or schools. And we've remained committed to that. I just want to mention that even for that period of a year, we were giving all those who tested positive through our PCR testing lab, it was given to them for free. Where do we stand now? We think, give or take how many yogurts have gone out there, Sankatana is responsible for over 4,000 recoveries. Now, there, Sahapi, you told us how much yogurt has gone into this community. What does Sankatana do once people have taken it? Well, practically nobody dies. That's a very important thing. Secondly, it shortens your disease period quite significantly. I mean, we have tested people who COVID positive, take Sankatana, and within 2 to 3 days, you test PCR negative. I mean, some of the stuff was overwhelming. It was like, what's going on here? There is a third thing that's going on. And it's this third one, which I think has been quite dramatic for us to grapple with. I'll perhaps unpack it in the following way. The third thing that's happening is that our yogurt is down-regulating NF-kappa B. Now, what does that mean? Perhaps I will just illustrate one story. I've got thousands of them, but let me single out this one story of an 86-year-old man who can't talk, can't walk, has difficulty breathing. He is literally on death's door. When we give him Sankatana 3 days later, his symptoms are gone. And you might say, wow, that's fantastic. I'll tell you what, the most fundamental thing which shocked me was when I was told that this old man no longer walks with his cane. Now, come on. He walked with a cane for 5 years, had COVID, was completely immobilized, was on the floor. 3 days later, boom, he's up, and now is back at his hobby of tending to his vegetables. I know, it sounds like the stuff of fiction. It sounds like the stuff where I keep telling David that what we've unlocked here is really, we're trying to bring you guys with us, but we know it's a difficult journey because it's difficult to believe, but we're bringing you to the cusp of where the remedy meets the miracle. I know, difficult terrain for scientists, or most scientists when you start talking miracles, but I don't know when last people were raised in 3 days. It sounds like something I read in the Bible. But anyway, that's what's been happening. And I'll say this. Why are we talking to you guys? You are indeed a tough crowd to convince. You are the most questioning, the most skeptical. In a word, I would say, or in a phrase, I'd say, you're tough to want. You play tough to want. But it is an intervention made at your level to convince you, which will make all the difference. Primarily because when you have the weapons we've developed in your grasp, not only will it help save your lives and keep you healthy, you then will be in a better position to save those who then come into your care. And look, this yogurt's done amazing things in Lesotho. And guess what? It was co-birthed in Lesotho and right here in Hilton. It's about time that it comes and does the same things here in KZN that it's done in Lesotho. We need you to hold hands with us because we can't do it alone. We're a research and development company. You heard Joey bombard you with science. That's what we do. But guess what? Sick patients come to you. You're the ones who are at the front line. This is where we've done our bit to try and inform you on how to fight this disease. We're trying to arm you with the weapons that we've developed. And make no mistake, Liselo Labs has basically made a laser-guided missile that's locked on COVID. And we're basically saying to you, if you are a pharmacy, we're happy to give it to you on consignment. Make sure that the customers have access to it. If you're running a COVID ward, we're happy to talk to you about donating it into your COVID ward. The most important thing is to get it down the throat of sick people, because it is then that you will observe the miracle that we've seen for the last year happen before your very eyes. So I challenge you and I say, knowing what we've told you now today, what are you going to do? Because I promise you, you will revisit this moment, and it will be one of, when you now know what you know, what did you do? I can promise you now, I've told David, well, guess what? People are dying. I know we're trying to patent, but I'm not going to sit idly by when I know what this thing is doing. Okay? So that's why we're here. The patents are a process, but we're about the business of saving lives, and we need you to help us in that regard. I leave you with this. For 150 rands, imagine this, nobody needs to die. Ladies and gentlemen, I've got this theme of the Avengers here, so I will basically say, from the Sutu I'll give to you is this vibranium. And my parting shot is essentially to say, Wakanda forever. I thank you. Please, questions and answers. Thanks.
Can I just ask if we give more speeches around? Thank you so much to Sehapa, David, and Joey. I'd now like to open the floor for questions. If anybody has a question here, you can raise your hand. We will also tend to those on Zoom. Any questions?
Right, so Dr. Gilbert here, obviously I'm a general practitioner dealing with sick patients, but also I do the COVID testing station. My question is, where do we get this? How do I get it to my patients?
So we're going to give you a whole lot of free samples now. You take as much as you want, please. We have Mandy here, who I think we've given probably a few hundred by now, that she's been giving into every single sick person she can find, and she will tell you a million stories about the recoveries, dramatic recoveries. So please, whatever you need for sick patients, give it to them. We want you to experience firsthand that it's rubbish, or it's indeed a miracle that we claim. Because like I said earlier, the clinical observation is the only demonstration of its efficacy that we can offer you right now. We're selling it here. We're selling in Spa, in Camperdown. It'll soon be in all the spas in the country, and a whole lot of pharmacies as well.
Hi, I've got 3 children. Is it suitable for children to take it if they have symptoms or get sick with regards to it, because they're not being vaccinated? So we need to know if it will be safe for them to have it, and maybe implement it into schools and things like that, where there's problems there with kids getting COVID.
Before you, can I ask how old are your kids? Can you just like to share?
One is 21, and the other 2 are 11 and 12.
I've dosed my 3-year-old with the syrup. He loves it. Yesterday we dosed White and Kosi's 2-year-old who wasn't sleeping well. Look, the honey-based one is honey, it's egg, and it's turmeric primarily. So there's some allergens there, which some pediatricians will be concerned about. But the honey is peeled honey. It's certified free of Clostridia. So all the normal concerns around honey shouldn't be there. We obviously consult your pediatrician, but we think it's safe for for toddlers, neonatals even. In fact, yeah. And just to the point of the schools and the vaccinated, obviously we promote get vaccinated. It's your best defense against severe disease. The fact of the matter is, in this country right now, we're sitting with 75% that aren't vaccinated. And those in the townships are part of where the so-called anti-vaccine thing is happening. It's not just the well-educated, supposed, alternative media crowd. It's the guys in the township that are also battling. So get vaccinated, number one. But if you're not vaccinated and you get sick, there's a therapeutic which we offer.
Can I just add that it's also safe for pregnant ladies, and for the Indian community, it's halal friendly. Thank you.
There we go. We have another question.
Can I ask a question from Zoom?
Cool. I'd just like to know the recommended dosage and for how long do you recommend taking it?
Would you like to take that?
Basically, it's 1 spoon a day for a 5-day regime. The really cool thing is there's no overdose. So depending on the severity of the symptoms, you can double it up and give them another spoon or 2 in the evening. You know what the biggest challenge is? Nyasha, can I please have that? Yeah. The biggest problem with this strawberry flavor is that it tastes so amazingly nice. People can't stop eating it in one sitting. That's the challenge. Yeah.
Please come close to the camera.
Which camera?
Yeah, we can't see you.
Oh,
Yeah, we
sorry.
can now see you.
All right, yes. So I've got the yogurt. And essentially we've got 2 flavors. We've got the plain, which is obviously compatible with people who have diabetes. And well, everybody else can eat the plain as well, to be honest. But if you've got diabetes, obviously stay away from the yummy flavor, strawberry. Right.
Okay, we have a Zoom question.
Okay.
Okay, you can ask. Dr. Brookman, you can speak. You can ask your question.
Oh.
Okay, thank you. I just wanted to know if you had done any work on your mice models with pulmonary lavage before and after infection, and with the use of Sankatana in mice. Does it work? Are there changes that you can determine objectively?
So thanks, Dr. Brookman. We haven't done our own mice models because for various reasons. Ethics challenges around getting clearance takes a while in South Africa. But others have done mouse model work on our active ingredients. And what is important around the mouse model work is that it's demonstrated a cytokine stabilization in a mouse model, the active ingredients. So when you provide the mice that are that have an induced immunopathology, when you provide them with the active ingredients per oral, they are showing a systemic restoration of the activation of NF-kappa B, or the dysregulation of NF-kappa B. And why it's relevant, I think, to the audience is that if you understand the role of NF-kappa B in disease, you cursory Google of NF-kappa B and whatever disease you choose, malaria, HIV, brain cancer, aging, okay? If you understand the link between dysregulated NF-kappa B and those pathologies, you understand the potential broad spectrum of application of an active ingredient that is in a yogurt, which can manage or mitigate a dysregulated NF-kappa B levels in a sick patient. So those mouse models have demonstrated that. We are trying to build the clinical data to demonstrate that in our patients. right? Where we can monitor the effect of NF-kappa B activation of cytokine levels, IL-6, IL-1, TNF-alpha in patients pre and post giving Sankatana. We are still stuck at ethics application stage in Lesotho and South Africa to start that work.
Thank you.
We have another question.
Oh, hello, my name is Siabonga. I work for Lancet Laboratory. What I want to know is, based on the study that was shown on the screen, I felt like to me it was not detailed enough. I wanted to know something more on Lesotho. This thing has been tested on Lesotho. You tried it on Lesotho. What's the COVID? I know COVID, it kills people. But mostly, there I've seen, based on my knowledge, people who are old, they die. People who have comorbidities, disease, they die. But to people who are young, who does not have comorbidity, it's a flu. They take treatment. After a few days, they are okay. That I noticed, but based on my knowledge. So now, those people who are taking this yogurt, were they not taking the medication? Does it replace the flu medication? Because flu medication, just people, they get fine after this. So on your stats, how many people were old? And how many people had comorbidities? How many people were given this thing? Because out of so many people that were given this yogurt, and then there were people who were old. There were people who had comorbidities, and they all recovered. They were not taking medication now or not. Thank you.
Okay, great question. One of the biggest challenges we've had is that we were pretty much given this to anybody and everybody who would test positive. Okay, so it cut across old, young, and with comorbidities, etc. Hence why, of the many thousands of examples, I selected the one of an 86-year-old man, okay, to show you that here's an individual who's definitely old and was looking like he was on death's door. We can share some of that data with you. We're still analyzing a lot of it with our US-based partners, because we want to then give out information around of X number of people who were old and of X number of people with diabetes, what was happening. What I can broadly say, okay, the most important takeaway is that nobody was dying, even in cases where we expected people to die. You know, when you're now administering it in a COVID ward, the nurses there have an expectation that these people who are now escalating to ICU, X number will die. I promise you, once they were on Sankatana and it was given to them in the regime we prescribed, they didn't die and they recovered quickly. I mean, everybody is in awe. I don't know if I've answered your question. I don't have numbers per se right now, but we can certainly make those available to you if you're interested.
What we've done is we've created a lot of testimonials, because the challenge with collecting data is that you need ethics approval to start the study, right? So collecting clinical data and then publishing that data, the immediate question is, where's your ethics clearance for starting the study? So we still have that ethics clearance process. So what we've done in the meantime is to collect all the testimonials, all the anecdotal data from all of these patients, not all of them, many of them. Some of them are video recorded, some of them are just text. And on that basis, you can get some idea. But like Saba mentioned earlier, this is just a fantastical story. It's mythology, okay? We're claiming things which are just ridiculous, which is why we want you please to take the yogurt, give it to someone you know who's sick, and monitor them 30 minutes after you've given them the yogurt. Ask them how they're feeling. Because we're noticing change in symptoms in 30 minutes. That's how dramatic the change is. Someone is coughing, someone has got a headache, someone's got fever, they're noticing an amelioration of symptoms within 30 minutes.
But I would just want to add, just so that your expectations are not built on David's 30 minutes. You will see or notice a lot of severe patients, a significant change in the state of illness, typically 24 hours later, because that's typically what would happen with patients who were bedridden, okay, bedridden, who, after having taken Sankatana, the following morning, they do things which their relatives wonder what's going on. They are up, they're cleaning the house. Do you understand what I mean? It is just phenomenal. There was a grandmother who they were looking for in a bedroom because she'd been out for 5 days. When they went and checked up on her, hey, flip, she was out on the stoop, enjoying the sunshine for the first time in whatever period she'd been down for. Thank you. Any more questions?
I would like to go off the subject now of COVID, and I hope it is not wrong to do that. I had a virus 20 years ago when I was still running, and I was 7 months in the hospital. Since then, I've got IBS and a very, very weak immune system. Will this help for me personally?
Elsa, a great question. And my short answer is this. It can't do any harm, and there's a good chance it might just help you. We would want you to give us feedback in terms of how it behaves with you. We really are keen on gathering all that data. But remember, as David stressed, it's a food. It's food. At the end of the day, it's food. We're doing something miraculous here. We're performing a miracle with food. Christ with 5 loaves and 3 fish. I feel it's there. I mean, it reminds me of a joke with a guy in Camperdown. His name is Basil. Vic and I were going down there to deliver about 100 odd Sankatanas when Basil said, "Oh, it's you guys with the Jesus yogurt." Clearly, obviously, the word had spread. And I quickly retorted, yeah, 1 drop of this and you can turn water to wine, and 2 drops, you can cure a leper. He then retorted, drink 2 tubs and you can walk in water. I promise you, it's probably the walking on water one which we're next going to test. So my short answer really, ma'am, is give it a try and let us know how it behaves. Because I can tell you countless stories. Take, for instance, an Indian lady walks into one of the pharmacies. Now, when I went to that pharmacy, I told them exactly the same thing. I said, look, I'm going to tell you something you're not going to believe. Anyway, tables were turned and the pharmacist calls me up and says, listen, Saab, I'm going to tell you something you're not going to believe. And I'm like, what? Shoot. She goes, Indian lady came in here. Her mother is a stage 4 cancer patient. She's got this, she's got that, and now she's coughing. Guess what I did just yesterday? Where typically I would pick any number of remedies that they have on their shelf. All I did was I handed them a tub of your yogurt. And the Indian lady said, yogurt? And she said, yeah. She promptly paid, and she was told, listen, come back in 5 days and tell us how your mom is. Guess what? She says she spots this Indian lady the very next day in the shop. So she walks up to her and says, what happened? How's your mom? She's like, listen, I'm here to buy 5 more tubs. My mother is a different person. Now, I don't know how to quantify that. All I can tell you is this. It literally feels like when I was a kid, I used to get excited about pulling carrots out of the soil because I never really knew how big the carrot was. You know, right now, our first attempt at pulling this carrot out, we realized, hold on, we're actually stepping and standing on the surface area of the carrot. We have to step back and try and excavate. We are literally on the tip of the crab of what this yogurt can do. And your experience will guide us in terms of just how big this carrot is. Thank you.
If anybody has any other questions? Okay, so I'd just like to thank everybody who attended in person and those on Zoom. If you have any other questions, please feel free to email them to me, or if you've asked them in the Zoom chat function, we will respond to them. But yeah, if anybody has any other questions. Okay, great. Thank you so much all for attending. And please come by and collect your free sample from us. Thank you so much.
Thank you. Cheers.